Hello and welcome to CROI 2015. We're here in Seattle this year. My name is Matt Sharp. I'm a HIV education and advocacy consultant and long-term HIV treatment activist, and I've been following the cure research parallel pathway for many years now, and we are here today with two extraordinary researchers who are hopefully one day going to lead us to a cure for HIV. We have David Margolis from the University of North Carolina and Louis Picker, who is from the Oregon State Health.
Oregon Health and Science University.
Sorry. And so it's interesting, these gentlemen are both, David is a principal investigator with the DARE collaborative CARE collaborative in North Carolina. I knew I'd get that wrong, and because I always do. And Louis is actually where you're working with the DARE collaborative.
Working with the DARE as well as other programs to work on cure.
And what's really amazing about this setup with this NIH grant named for Martin Delaney is that it's a collaboratory effort, and it brings scientists together. They're really doing work together in many ways already, but it's a systematic approach to getting people to collaborate, and it's yielding some very interesting science these days. And so we're here at CROI, and there is a lot of sessions on where we are. I thought we would start just with giving a little brief, very short introduction about where we started and where we are today, and just to give people who maybe are not aware of what the science is so far. And then we'll cover and talk about updates. So, David, you want to start?
So I guess when we're talking about trying to eradicate infection, we start at the place where we are currently with current therapy, which restores health, maintains health, can make people so-called aviremic. That's undetectable levels of virus in their blood on routine testing, and that's a stable state that can be maintained by treatment, by drug therapy for many, many years. And of course, the limitation of that is that it requires health systems that deliver people medical care and medical therapy for many, many years. So that's a great result in a lot of ways, but maybe not what we want to have for the future of forever and ever of mankind. So to seek something better, people have over the last few years started to explore, and more recently, as you say, I think in a much more rigorously focused scientific way, ways to reduce the virus that persists in the body despite therapy, that is not eliminated by current therapy, but is only suppressed and controlled by it, and exploring different ways to either remove or eliminate all the cells that can make virus, or to control that in some way that therapy can be stopped. And that at the same time health and non-infectiousness can be maintained. And so there are several different sort of paths to this goal that have been developed, and we can both talk about these different pathways, and I don't think anybody knows how we'll eventually get there. A lot of people, I think, suspect that many of the approaches will merge and overlap and be combined in the future. But it is very early days. Over the last couple of years, we had some of the first studies showing that giving a drug, giving a medication to a patient that was taking therapy and had undetectable viral load, undetectable viremia, that giving a specific kind of drug could disrupt latency or wake up the virus that was hidden. And that was the first clue that we could maybe use that as a tool to move us towards ways to get rid of those cells that can produce infectious virus. We could talk about some of those sorts of things that were at the meeting. There were, I think, two really larger sessions that sort of went over two aspects of that work. One was really a session, a lot of different investigators. I can't even remember all of the details and all of the people, but it was essentially presentations of different studies trying to actually measure how much virus was in cells and what were the best ways to measure these very rare events of cells that were infected with a viral genome that could produce an infectious virus.
And this is kind of a big area of contention right now because there's a lot of different ways to do that, and the way to do it best is still yet to be.
Well, and I guess what really was well illustrated at the meeting by all of these presentations was that the biology of this is quite complex.
Right.
There are many cells that a virus has entered and has gone into the chromosomes, into the genome, and there's a piece of viral DNA there. But most of that viral DNA actually turns out to be in one way or the other junk. But inside that mountain of junk DNA is a problem virus that's really there. And the question is how to measure which cells have really the virus that we have to worry about in the noise of all the cells that were infected, but that's not a dangerous virus because it's not working or defective or mutated in some way.
So can you tell us where we are with coming to that agreement of that measurement and as of this point? Has it gotten any further, or has it gotten more confusing?
I think it's gotten further, and I think that the studies that have been done over the last year, some of which were presented at the meeting, just showed how complicated it is and how difficult it is. And that this will require really a lot of work to be able to have measurements of how much dangerous virus is really left.
Right. So let's switch gears a little bit and talk about what you're working on in terms of the immune system, in terms of vaccines, and then we'll come back and talk about a little bit more about the specific drug approaches.
Yeah, and just to pick up from where David left off is the concept is to sort of induce this latent virus into a form where it expresses viral proteins, and hopefully you can kill those cells. Now, the initial hope was the cells would just sort of kill themselves because the virus would kill them, but that clearly is it may be some of the times the outcome, but often is not the outcome. And then there's even the possibility that cells can actually that are infected with the virus can divide and maintain replication competent virus that can start a new infection. So the latent reservoir can actually respond to normal lymphocyte homeostatic mechanisms and increase the reservoir by virtue of cell division. And one of the interesting things that came out of the conference so far is that you can get what they call these clonal expansions with clonal virus that is replicated in different cells because the cells divide, and that some of those at least are live virus that can potentially reactivate infection. So the immune system is intimately wrapped up in all of this. So on a simple level, one, we have to make sure that the cells that we awaken the virus in, that the virus is awakened by the treatments, their latency reversal agents, die, because you want to get rid of those cells. One possibility is they'd kill themselves. Another possibility is that they're recognized by the immune system, and the immune system kills them. And so, the first sort of barrier is to get those cells activated so they can be recognized in the immune system, and then the second sort of barrier is to figure out how to get the immune system to recognize. Now, in a paper that came out a couple months ago or a month ago, one of the problems is of course, individuals who have been put on antiretroviral therapy after about 3 months of infection, the virus has already escaped their own natural immune system, their own immune response. So they've lost
that ability to.
So the immune responses have a very stereotypic way of responding to viruses, and everybody's a little different. But the virus that's in those people have escaped. They basically mutated their genome so that you're no longer recognized by the antiviral T cells, and that's what comprises the majority of the reservoir. So it already suggests that to use T cells, cytolytic T cells, to kill the reactivated cells, you have to come up with something different than the natural response, and that means focusing on areas of the virus that haven't mutated. or certainly areas that haven't been recognized by the initial response. And that's some of the work that I'm doing is trying to develop vaccine vectors that generate T cells that recognize parts of the virus that haven't escaped, so they can still be recognized and destroyed by the immune system. So in a sense that we already have a two-step process. You got to awaken the virus with the kind of agents that David was talking about, and then you have to have that awakened virus, those cells killed or otherwise done away with by the immune system. And then, and then it turns out there's actually a third barrier, and that sometimes the virus awakens in places where the immune system can't get to it. And
right.
We just had a paper published on that the B cell follicle actually is one such place where virus can persist and in fact even replicate and spread in a condition where CD8 T cells can't get to it because they're excluded for normal immune physiologic reasons from the follicle. So we call those the sanctuaries. So you have, the barriers that we have are, first of all, the latent reservoirs, second of all, killing the cells that you awaken, and third of all, getting around those sanctuaries.
And the CNS is another.
And CNS is potentially another, and there may well be others. And the important part is identifying these barriers so that then you can develop approaches to get around them.
Sure.
And we're sort of at that barrier identification phase. I think that we're already seeing that people are beginning to say, oh, well, that's a barrier that we can get around this way. One of the things I was going to mention is that one of the advances in recent years has really been the availability of highly active antiretroviral therapy for SIV in monkeys.
Yes.
And that allows us to a couple things. One, it allows us to go in and really look at what's happening in the tissue, which you're sort of precluded from in patients to really go down and look at the tissue reservoirs.
Right.
And the second aspect of it is it allows you to do experimental manipulations that can affect the barriers in one way or another that would take you a long time to do in humans, even if it were possible due to safety concerns. So
right.
I think that has just come online in the last year, the ability to do these kinds of reservoir studies and reservoir immunobiology of the reservoir studies in monkeys, and I think that's going to really lead to a lot of new insights.
And that's some of the work you're doing.
And that's that's my primary focus is on the monkey model.
Well, thanks for explaining that to me. That's the best explanation I've had of that problem that we're having with that pathway. So, but you want to explain where we are exactly with the HDAC inhibitors and maybe some of the combination approaches that you're talking about.
So I think that I was going to perhaps highlight some of the things at this meeting.
Okay.
So certainly there have been studies over the last couple of years of perhaps what would be the first class of latency reversing agents, these drugs called HDAC inhibitors. These are drugs that affect an enzyme that is in every cell that helps regulate genes, and using this inhibitor of the enzyme, one can see modest changes in host gene expression and some increase in latent virus expression. And so there are now several of these kinds of HDAC inhibitors that have been tested in various cell systems in animal models and even in some people. And there's pretty clear evidence that at least it's a step forward. There's some evidence that you can disrupt latency to some level with these drugs in patients in clinical studies. Really the critical unanswered question yet is that how many of the latently infected cells are really revealing themselves to make an antigen, a protein, a piece of a virus, so that they can be targeted by an immune response and cleared. And in sort of answer to the concern that the single agent, the HDAC inhibitor alone, may not be good enough or effective enough, in a lot of work in a lot of places have started to discover and test other kinds of compounds that could work with the HDAC inhibitors or in other combinations to do the same thing. And I guess probably one of the most interesting presentations at this meeting was some work coming actually out of hepatitis B research, a compound that was developed as an adjuvant for the immune response of hepatitis B to sort of tweak the immune system, but given to an SIV-infected monkey, induced some activation of virus in the blood in a monkey that was suppressed on therapy. So we don't actually know exactly how this compound is working, whether it's working directly on the latently infected cells in the monkey, or whether it's a sort of second level effect where it's causing an immune activation, and that activation leads to expression of virus, but it's a new tool to sort of start this process going. So I think it's another important sort of step-by-step building towards the ability to have these combination approaches.
And what was that compound?
There was a compound that's called GS, some
Yeah,
number
999.
Does it
We
have
call it
some
TLR7 agonist.
Does it have
It's the class of drug. It's an orally available
Right.
TLR7.
So it binds to and activates a receptor that's found on some kinds of cells in the immune system. And this compound is a Gilead compound, but actually there's a variety of these compounds in development in other places.
And so this is at this meeting is actual step forward for a new target. So that's good news.
Well, it's very good news from both the therapeutic standpoint is that clearly we're getting a more potent latency reversal agent, but from an experimental standpoint, it's also good news because now it gives you something to target. For example, in my work, now I have a compound that I know that can reproducibly reactivate a certain fraction of virus, then I can begin to ask questions about what immune responses can stop that, or begin to use that as a tool, that compound as a tool to better explore the next step, which is the.
So even if this particular drug turns out to be too toxic to use in people or whatever, it's another way to move the field forward.
Yeah, you'll learn a lot from even the mistakes that might be made from the development of it. Yeah, that's exciting. It's exciting news. So I think we have a little bit of time. I want to just, if one of you could just say where we are with the gene therapy pathway. I know that's not necessarily your fields, but just so that we can inject that into this. There wasn't really much at this meeting that I saw.
Are you talking about the antibody or the entry inhibitor type gene therapy?
Well, either one, but that towards the cure or remission or whatever we're talking about. Can you state where we are, if there's any new progress at this meeting?
Well, there's lots of different kinds of gene therapy approaches. A lot of them are in the phase of that certain kinds of gene therapy tools are available and can work in the lab or in a model system, but to really be able to move them into animal models and people in an effective way is sort of the next big hurdle. I think it might be interesting to talk about this sort of synthetic antibody that was shown.
Briefly.
Yeah, that's sort of the general topic as people are now beginning to explore. Now that we have antibodies that can broadly recognize neutralize viruses, that means they can broadly bind viruses. You could use those as tools to look at cells that express envelope on the surface and maybe kill them. And so both in terms of just giving the antibody and in terms of doing gene therapy in which you actually have the antibody produced by the person inside their own muscle cells, those are both sort of congruent, different delivery methods for the same approach. I think data presented at this meeting today suggested that antibodies alone, and antibodies sort of bridge the gap between therapy because they definitely can prevent viral spread and potential cure therapy because they could potentially, although it hasn't been very well demonstrated, kill infected cells. That they sort of blur that distinction. But it seems that by themselves they don't cure monkeys. Let's put it that way. And maybe they have some beneficial effect, but whether that's due to the fact that they sort of enhance therapy in the sense that they make viral replication spread even more suppressed, or whether they're actually killing cells still hasn't been shown yet. But that's the promise that you don't need. It may not be just only T cells, cytotoxic T cells that can kill the activated cells. It could be antibodies. Now the downside of antibodies is that they only see things that are on the surface of the cell. So there has to be not only reactivation of viral gene expression, there has to be actual viral production and surface envelope for those cells to be seen, whereas T cells can see even the earliest production of any viral protein. And in fact, another interesting talk was today about the fact that the untranslated 5' leader sequence of viruses that are integrated in the genome gets translated and elicits an immune response that actually can cause immune escape. And so that's exciting because that's present in all transcripts that are made by the virus, and potentially could be something that happens very early, or even, potentially, we don't understand reactivation. You could have reactivation all the way to making viruses. You could have reactivation that just makes a few viral proteins, and anything in between. But this 5' untranslated leader sequence, if it gets translated and put on the surface as peptides the T cells can recognize, it would be a very early sign of an infected cell that a T cell could recognize. So these are so probably both antibodies and T cells can make a contribution to the killing part of the reactivation. And the gene therapy part is really just the fact is, you don't want antibodies have a half-life. You'd have to keep giving them but if you actually take the gene for the antibody and put it in the muscle, then it'll be continuously made. The downside of that is that you can get the immune system may eventually recognize what's made, and you can't turn it off very easily. At this point, you can't turn it off. So
that's the scary part.
So there's some scary issues that need to be overcome, but it's
so it's the beginning. It's the beginning.
We're in very, very early stages, and stay tuned for next year's conference, and maybe we'll have some bigger updates for you. Thank you for watching.